Okay, now that we have talked at length about DNA copying itself, DNA coding for proteins, transcription, translation, our last piece of our unit on molecular genetics, how genes work, is learning about stuff we can do with DNA, with the technology that has developed around our knowledge of this molecule. Um, so, doing stuff with DNA is really a discussion about biotechnology, and that's the uh, set of tools we use to work with DNA. Okay, so that's what we're going to talk about with this, and this is going to lead us into our labs that we'll be doing with this unit as well. All right, so what can we do with DNA with this knowledge? Well, we can do really weird things that seem to have no purpose, at least in and of themselves. Turns out sometimes they do have a purpose. But we can genetically alter animals, which we've done. You can see we have here this, let me get my pen. This mouse right here, which is glowing in the dark. We've got this pig that seems to have glow in the dark aspects of his anatomy as well. We have featherless chickens, and if that's just a really bizarre looking thing right there, but featherless chickens that have been engineered. We have plants. These are grains of rice that have additional nutrients because we can alter their genes. We have tomatoes that you have probably already eaten some genetically altered tomatoes. These are genetically altered tomatoes that stay ripe longer than their unaltered counterparts. We can analyze DNA and use it to figure out a suspect at a crime scene. We can even use DNA, genetic engineering, to create medicines that we need. So there's just tons and tons of ways we can manipulate DNA and use it to do things. Some people think maybe we shouldn't be doing some of the things we're doing, and that's certainly a point for ethical discussions, but what we're going to do at this juncture is simply talk about what things we are able to do and what things we might be able to do in the future. Okay? Now, to put this in some perspective, you need to understand that manipulating genes to do things that we want is really nothing new. Humans have been crossbreeding or selective breeding for years, and all that means is you pick critters you like that have traits you like, and you breed them. And obviously the goal is to get babies, offspring, that you with traits that are really, really great. And that is how we have arrived at all these many breeds of dogs that we have. Okay, as shown in this diagram. Now, selective breeding or crossbreeding, same thing, does have its limitations. You can't cross just any old things, okay? In other words, that cross right there is not going to happen. You are limited to staying within your species. Whereas if you are using the tools of biotechnology, you are not limited to just crossing within a species anymore. You can take genes from an entirely different organism altogether and put them into a different species. Pretty weird stuff. Additionally, cross or selective breeding is not terribly precise. Things don't always work out like you intend. Maybe the traits that you thought were really great in this one organism and the traits that you thought were really great in this other dog, you know, they don't mix together in a way that you think they should. Yeah, we'd hate to see something like this guy pop up. So, anyway, so you may recombine traits that weren't necessarily intended. Today, doing stuff with genes generally means, though certainly crossbreeding is still done, don't get me wrong, but we are in this class going to be talking about biotechnology. biotech is essentially a set of tools that allows us to much more precisely manipulate DNA. We can actually pick genes we want out of one organism and put them in another, or we can really analyze the genetic code of an individual's DNA to learn about it, to learn all kinds of things. Okay? With biotech, usually there are fewer unintended results than when we just cross organisms and hope things work out. Though some folks do argue that there may well, in fact, be huge unintended results for our environment if genetically altered plants start cross-pollinating with other plants in our environment. We may create things that we don't intend, and so on. So there is a lot of debate surrounding this issue. At any rate, if biotech is really a set of tools, then what tools is it? What tools do we need in order to manipulate, to do stuff with DNA? We need tools that allow us to cut DNA into pieces. We need to cut DNA into pieces to analyze it, to cut it out of a cell of one organism and place it into the cell of another organism. We need to be able to cut DNA fairly precisely. We need tools that allow us to analyze those things that the DNA fragments we have cut, figure out what we've got. We need tools that will allow us to select certain bits of DNA that we want to use. We need tools that will allow us to put DNA into other organisms if we want to genetically alter other organisms. And we need tools that will allow us to make lots of the protein product that a particular gene codes for. One of the reasons we might want to focus on one particular gene is we need the gene product the protein product that gene makes, maybe for a medicine or something like that. So we have to have ways of doing that as well. We may not use all of these in the same instance, but for different purposes, we need these different tools in biotechnology. Okay? Now, before we can do anything with a DNA sample, it does have to be big enough to work with. And sometimes we have a very small DNA sample and we just need to make it bigger. Bacteria have, generally speaking, been the workhorses of the biotech toolkit for years. They have allowed us to make large samples of DNA from a small sample, and they've allowed us to make large amounts of protein product from a DNA sample. So we're going to start by talking about bacteria a little bit. Let me get out of this. Okay. All right, and let me pop over into this. All right. Bacteria have been really, really important in allowing us to work with DNA in a meaningful way. So let's talk a bit about bacteria and what they're all about. And this will help us in lots of aspects of our knowledge of biology, but certainly biotech. Okay, remember bacteria are prokaryotic. They are single-celled. They reproduce asexually. If you're a bacterium, there is no sex for you. They reproduce by something called binary fission, which is probably the ancestor process to mitosis, but they are very different. Binary fission, if you'll recall, a bacteria, that's a bacterial cell has one DNA molecule in it, and that DNA molecule, while it is a double helix, because that's what DNA molecules are, that molecule is in a circle. It is not a single string of DNA like we see in our eukaryotic cells. Okay, that's not what we've got in DNA. Whoops, I'm sorry, in bacteria. We have this single circular chromosome. And essentially, in binary fission, what this thing does is it copies itself, so we end up with 2 rings of DNA in the cell, and then the cell just cuts itself in half. And that's it. It's pretty simple. And it takes about 20 minutes. That means bacteria can grow really, really fast. They can reproduce really, really fast. Overnight, you could get from one bacterium a colony of 100 million overnight. That's how fast they reproduce. In terms of sheer numbers and their diversity, they are the dominant life form on Earth. We don't really think of them because we don't see them when we walk outside or wherever, but certainly they are incredibly, incredibly successful, even though they are also, compared to us, very, very simple. Okay? So this is looking at that bacterial genome. It's genetic information we were just talking about, a single circular chromosome. It is naked DNA. What that means is the DNA in a eukaryote, because it is so long and stringy like this, gets easily tangled. So in eukaryotes, we have proteins called histone proteins that we use to wind the DNA molecule around, just like thread around a spool, like that. And so this in the center might be the protein, the histone protein, and this is the DNA. Okay? We don't have that in the bacterial genome. It's really small amount of genetic information in a bacteria, and as you can probably imagine, about 4 million base pairs compared to many, many more in a human. I think, oh, gee whiz, I had a number. It's certainly in the billions. About 4,300 genes. So hugely, eukaryotes have considerably more DNA than that. How then, since they are so genetically simple, have bacteria become so diverse? This is important for understanding how we can manipulate them. Okay? Experiments have been done with bacteria that show that if you mix bacteria that have been killed, okay, they are pathogenic, meaning they are disease-causing, but we kill it. And we mix these in a test tube with some non-pathogenic, non-disease-causing bacteria that are alive. So these non-disease-causing are alive, these disease-causing are dead. But we mix them together in a test tube and put the mixture in to some mice. The mice end up dying, even though the disease-causing bacteria had been killed. So, what has been concluded from that experiment is that live, harmless bacteria must have picked up something from these dead bacteria that transformed the harmless ones into something bad that eventually killed the mice. What is it? The question is, what was, in that experiment, the transforming agent? What was it? Okay? It was DNA. It was determined through experiment that what the bacteria that were alive but harmless had picked up from the dead but harmful bacteria was DNA. Perhaps DNA from those harmful bacteria started to come out of their dead cells, okay? And these live bacteria are designed to be able to pick up DNA from their environment. Wherever they find it, they can pick it up from other bacteria, from just laying around, whatever. And then they will incorporate bits of that DNA into their own circular chromosome, and then they can express those new genes just as though they had always had them. This process is called transformation, and we can use this process to our advantage. If we can throw some DNA into a test tube with some bacteria, they tend to pick it up pretty easily without us having to do a whole lot to them to make them do that. And then they can take on the characteristics that that DNA codes for. This is a form of genetic recombination. Recombination just means combining new sets of genes. Your parents did some genetic recombination when they created you. Dad's genes combined with mom's genes. Well, this is a different form of recombination. In this case, a bacterium might pick up some DNA from another bacterium or from its environment, just laying around, whatever, and then it expresses those traits. Okay? Now, this is an important word. In addition to the main DNA molecule, which is shown here in this picture. This is the double helix, the main molecule of DNA, the chromosome in a bacterial cell. There are other smaller pieces of DNA that are indicated by these smaller blue rings in this bacterial cell. They are called plasmids. Each of these is called a plasmid, and they are simply small supplemental pieces of DNA. They have maybe as many as 20,000 base pairs. Very, very small compared to what's in the main genome. Maybe much less than that. But they're tiny little circles of DNA. They copy themselves just like the regular DNA does, but they carry supplemental genes, extra genes. Maybe as few as a couple, maybe as many as 30. It kind of depends on the plasmid. But in bacteria, often what these plasmids are carrying are genes that make the bacteria resistant to antibiotics. Antibiotics are chemicals that occur in nature that kill bacteria. Fungus produce them naturally, and that's in fact how antibiotics were discovered. Now you get a prescription for antibiotic if you get sick with a bacterial infection. Okay? But these little plasmids can be pretty easily exchanged between bacteria. It's not exactly sex because bacteria don't have sex, but these plasmids can be released into the environment and other bacteria can pick them up. And this also aids in the very rapid evolution of some bacteria. If a bacterium here is his DNA molecule, maybe he's got some plasmids, but maybe he has not got an antibiotic-resistant plasmid. If he picks up one of these plasmids into his cell, and all of a sudden he's resistant to bacteria, he's going to have a much better chance of surviving and passing on these genes to its offspring than if he hadn't got them. And that can be a very rapid evolutionary advantage for that bacterium. Okay? And again, these plasmids can be imported just from the environment. If a bacteria dies, leaves its plasmids laying around, another bacterium could pick it up. Okay? Now, the question is, how can these plasmids, these little bitty molecules of DNA, help us? Remember, this whole talk is really not about bacteria, it's about biotechnology. Okay? So, plasmids are basically a way for us to get genes into a bacterium really easily. Okay? If we are able to put a gene we want into a plasmid, we can easily get that plasmid into a bacterium. So the plasmid is considered something called a vector. A vector is simply something that carries something into something else, and plasmids can do that. And then if we get that plasmid into the bacteria can express the genes that are on the plasmid, which basically means, as you know from our talk of transcription and translation, bacteria can simply make new proteins now. Okay, so here's what we would have to do. We would have to be able to get plasmids out of DNA, and we would have to be able to cut the plasmid open. We would then have to be able to isolate a gene that we want from another organism. Do you remember those glow in the dark mice we saw? Well, maybe we want to make glow in the dark bacteria. So if we can get a gene that makes things glow in the dark from another organism, can you think of any organisms that glow in the dark? I'll tell you one. Jellyfish. If we can get a gene from a jellyfish that makes it glow in the dark and insert it into the plasmid, okay? Boom. Okay, so here's our plasmid. We stuck in the gene we wanted, and now all we would need to do was get the bacteria to take up that plasmid, which they do quite naturally, and then that bacteria might make the protein we were interested in, the glow-in-the-dark protein. Okay? So plasmids are common or have been commonly used and are still to put new genes into bacteria. So here is like the gene we want. Maybe it's the gene for human insulin. Who knows? Okay, if you're a diabetic, you don't make insulin. Maybe it would be helpful to you to be able to take insulin. So there's the gene from a human for insulin. Maybe it's right there. Okay. Now what we do is we cut the gene we want out of the DNA. We cut and so there that is right there. That's the human DNA here. We cut that gene for insulin out of it. We open up the plasmid, cut it open, insert the gene we want into it, and then put that in the whole plasmid into a bacterium. And then guess what that bacterium can make? Insulin. Okay? So recognize that enzyme name, ligase. That would be used to knit those two things together so that they are one piece of DNA again. This would be called a recombinant plasmid. Okay? Now, the question is, how do we cut DNA? All right? And we're going to talk about that, but I'm going to take a break now since we've talked a little bit about bacteria and their importance. I'm going to take a break and we'll come back and do the second part of this podcast, which should be podcast 34. So thanks for watching. I'll catch you in the next one. Bye-bye.